this topic we're going to talk about today is genetics and heredity. And the information for this unit can be found in chapter 11 of your textbook. So we're going to start off talking about pretty much the founder of what we know about basic principles regarding genetics, and that is Gregor Mendel. So Gregor Mendel was doing his work in the early 1960s. He was a monk who lived in Austria, and he studied science and math, specifically probability and statistics. That was his specialty area, and he studied that at the University of Vienna. So he was a scientist who used the scientific method to come up with his principles of how alleles are inherited among different generations of plants and animals. So his primary organism that he worked with was the pea plant. So that's the garden pea. And what he liked about the pea plant is that it had some special characteristics, which we'll talk about in just a minute. But he did not have a microscope, so he wasn't able to look at anything at the cellular level. All he could look at was the phenotype, which is the physical characteristics of the plants as they developed from two parent plants. So he didn't know anything about chromosomes or cells. All he noticed is that when I put certain plant characteristics together in two different plants, this is what the offspring looked like. And he was able to repeat those experiments multiple times to prove that if I do this and this is what I'll get. So he worked again using the scientific method. He was a scientist who had a strong background in science and math. And he kept very detailed records of that, so he could, again, share his work and other people could repeat it, which is what we see is important for the scientific method, that the experiments are repeatable and you get the same result. So what he liked about the garden pea is it was easy to grow. When you talk about cultivate, it just means easy to maintain and grow in a lab setting. It had a short generation time, which means that once you cross-pollinated two plants, you could get the seeds and the embryos and a new generation of plants, the next generation, which we call the F1 generation. You could get that fairly quickly, so he could, run through his experiments rather fast and repeat them rather quickly to come up with his conclusions. So normally these plants can pollinate themselves, which means they can produce the same exact plant, but you can get genetic variation by cross-pollinating plants, which means you're taking the pollen from one plant and crossing it with another plant. So you have, we haven't talked about this yet, I don't want to get into a lot of detail, but plants have female parts and male parts, much like sperm and egg. They have different names, of course, but we'll talk about that later. But anyway, you can cross-pollinate two different plants to see how those alleles are inherited in the offspring. So we could call that, breeding among plants by having male parts and female parts creating new offspring. So the traits that he observed were simple and objective. For example, when we look at the different traits, here we can see in this chart, he found through his experiments that a tall pea plant was dominant over a short pea plant. Or the shape of the pea pod that formed, it would be either inflated or constricted. The shape of the seed was either round or wrinkled. Seed color was yellow or green. Again, by doing his experiments, he could see which traits were recessive and which ones were dominant. Because remember, the dominant trait is the one that we see expressed in the offspring. So if they carry the dominant allele for a round seed, we're going to see that expressed in the offspring if they inherit that dominant allele. So flower position would be if it's axial, like the flowers, here on somewhere along the stem, or terminal, the flowers on the end. The color of the flower, purple was dominant over white, and the color of the pod, green was dominant over yellow. So we had these great set of traits. What do we see? 7 different traits here that he could manipulate and even double up on and determine which one was recessive and dominant and predict the offspring. So he developed, his principles using what we call a Punnett Square, and we'll talk about that. So moving on then, he had crossbreeding, which again, he was taking plants that were homozygous, which means they were either homozygous dominant or homozygous recessive. And remember what that means is, if we use the letter T, capital T, for the trait being tall, So he would take a plant that had two alleles for tall, which would be homozygous, so big T, and he would cross it with a plant that was little t, and little t would be the short. That would be homozygous recessive for short, because we always use the letter of the dominant trait as our allele. So big T would be dominant, would be homozygous dominant, and little t homozygous recessive. So by crossing those, he could predict the offspring, which would all be heterozygous, and we'll talk about that. So we call that a monohybrid cross when we're looking at one trait. So if we wanted to just cross plants and focusing only on tall or short, that's a monohybrid cross. If I want to look at a dihybrid cross, then I would pick two traits. I would pick a tall plant with inflated pods, or maybe a tall plant with purple flowers, or a short plant with white flowers. That's a dihybrid cross. Di meaning two. So we'll start with monohybrid to kind of keep things simple. But some terms we use for the generation that are the parents, we call that the P generation. So we're looking at the first set of plants that we're going to cross. And then the offspring we get from that first cross, we call that the F1 generation, or the first filial generation. For example, you are the F1 generation of your parents. But now if we cross the children, so if we cross the plants that result from the first set of parents, if we cross those plants, then we get the F2 generation, which would be the second filial generation. And we can see how traits are inherited from the first set of offspring to the second set of offspring. Now obviously humans don't do that. We don't cross brothers and sisters to get an F2 generation, but we can do that in plants, and it helped Gregor Mendel come up with different laws of genetics, and one of them was the law of segregation. So the law of, well, let's take a look at this first before we get into too much information. So if I look at this first parent generation of a monohybrid cross, we're taking the homozygous dominant parent, in this case the tall plant, and the homozygous recessive parent, which would be the short plant. We know that when they form sperm and egg by meiosis, remember we have a diploid cell here, two alleles, but when they form sperm and egg, we only get haploid cells. So these gametes, the only gamete that this plant can provide to the offspring is a big T, because they only have big T's to offer. This plant can only offer little t's because that's all they have to offer. All their cells are little t. So when this, let's call this the sperm and this the egg, when these come together, the offspring are all going to be big T, little t. So we call this the F1 generation. All the offspring of parents that are big T, and little t are all going to be heterozygous. So they all are going to be tall because we know the big T is expressed and dominates over the little t. So even though they carry that recessive allele for being a short plant, all the plants are tall because they have this big T dominant allele. So this is the F1 generation, and every offspring of these parents will always be heterozygous, and they will look like a tall plant because they have that big T allele that they inherited from this parent. So when these offspring, when this F1 generation produces sperm and egg by meiosis, they're going to have a big T gamete and a little t gamete. And then if I take another plant from that same F1 generation and cross it, we're going to see another big T, little t sperm. So if we take a female part of the plant, and those are the eggs, we put those along the outside horizontally. We put the sperm vertically, but I could switch it either way. It doesn't matter. But I put the gamete possibilities out here on the outside. So again, this cell of this plant, when it undergoes meiosis, remember we get 4 haploid cells at the end of meiosis. We're going to get 2 cells that are going to have the big T, and we're going to have 2 cells that are going to have the little t by meiosis. So these alleles split. That's what we call the law of segregation, is they split. So we have these 2 alleles, possibilities, big T and little t. Those would be the eggs coming from this plant, and sperm coming from a plant like this would be big T, little t as well. So then we're going to cross these. So we're going to pretend that this egg and this sperm came together at fertilization, and it would form a big T plant. But if the little t egg was fertilized by big T sperm, we would have a plant that is big T little t, as we see here. If this egg and this sperm come together, again, we'd have a big T little t plant. And if this egg and this sperm came together, we'd have a little t plant. So when we do Punnett Square analysis, we're taking the egg and sperm possibilities and putting it along the side, which means we're just splitting the alleles from the parent. So in each case here, the egg and the sperm are coming from this F1 generation that we got from the breeding of these two plants. So we're going to take this individual, split the egg possibilities here, split the sperm possibilities here, and then we're going to fertilize by combining these two alleles. And this is our F2 generation now, because it came from parents of the F1 generation. This was the parent. We took plants from the F1. So what we see now is we have 3 plants that have inherited the dominant allele and 1 plant that has 2 recessive alleles. Now from going back to our chart, we know little t means that we're going to have a short plant. So I'm going to have, when I look at the phenotypes, which is what we see physically, what the plant looks like, we have 3 tall plants and we have 1 short plant. So the phenotypic ratio we say is 3 to 1. 3 tall to 1 short, because we have, each of these represents, 1 in 100, or 1 out of 4, or if this was 100 plants, each of these would be 25. We kind of talked about that in our lab that we did, for those of you that did the Tuesday genetics lab. So anyway, we have a 3 to 1 phenotypic ratio. If we look at the genotypic ratio, which means how many different genotypes we have 1 big T, we have 2 big T little t, or heterozygous, and we have 1 little t, which would be the homozygous recessive. So my genotypic ratio would be 1 to 2 to 1. So the way we write that in a monohybrid cross, when we're looking at 1 trait, the way we write that, I'm just going to pull up a Word document here to make it a little easier. So get nice and big here. So the phenotypic ratio in a monohybrid cross, can't type, we're looking at big T little t times big T little t is always going to be 3 to 1, which means 3 tall, 1 short. Now the genotypic ratio, looking at the same thing, I'm just going to highlight this so I don't. Is going to be 1 to 2 to 1, which means I'm going to have 1 big T little t, big T, 2 big T little t, and 1 little t. So those ratios are just expressing the different allele combinations I can have in that F2 generation. So this is the F2 generation. When I started out, remember, with the big T, little t alleles. So F1 generation, F2 generation, when these become the parents. Okay, so his laws from this observation of looking at these plants, taking a homozygous dominant, homozygous recessive, getting these all heterozygous F1 offspring, and then breeding those two together and getting this 1 to 3 to 1, or 1 to 2 to 1 ratio, or 3 to 1 phenotypic ratio, he developed the law of segregation. So he said each individual has a pair of factors or alleles for each trait. So an allele is just an alternate form of a gene. So when we define this, you should write this down. Anytime I'm writing something here, this is like I'm writing on the board. Write this down in your notes because it will appear on the test. An allele we can define as an alternate form of a gene. Okay, so big T or little t are examples of alleles. These letters, okay, so they're expressed as letters, capital or lowercase. Okay, so these are alleles, alternate forms of a gene. That's the definition of that. So going back then, so we know that these factors separate by meiosis during gamete formation, and each gamete has only one allele. So we saw that when we looked at this. This parent only produced one allele in its gametes. Again, sperm or egg, there's only one allele present. This has only one allele, so when we look at that, they come together by fertilization and form an offspring. Okay, so back to this then. So each gamete contains one factor, and fertilization gives the offspring two factors. And we saw that as well. When these came together, big T and little t, when these gametes came together by fertilization, the offspring now has two alleles for the trait. And whichever one is dominant is what is expressed, right? So when we look at these three again, we said they each inherited the big T, so they were all tall plants. This one only had little t, so therefore it was a short plant. So other things that he came up with his observations is the dominant allele is given a capital letter, and it always masks the expression of the lowercase or recessive allele. So something that is heterozygous in this case is going to look like the dominant parent. So phenotypically, when we talk about phenotype, that's the physical appearance of an organism, we can see these are the same. They're both tall plants. However, genotype is different. Genotype is the combination of alleles that we can't see but we know is there. So their genotype is different. This is big T, and this is big T, little t. So when you talk about genotype, we have a heterozygous plant here, and this we have a homozygous dominant plant. The genotype is homozygous recessive, two little t's. But the phenotype is different here. They look different. These do not look different. These are phenotypically the same. They both are tall. They have the dominant allele, so that's what they express. Okay, moving back then. So alleles occur on a homologous pair of chromosomes at a specific location on a gene. So we talked about that when we studied meiosis. If something is homozygous, they have the same alleles. If they're heterozygous, they have different alleles. So if we go back a little bit into our notes here, into meiosis, I'm just going to come back a little ways here. I can show you where we talked about that. Oh boy. Okay, right here. So again, these are homologous chromosomes. So these red ones here are homologous. Let's go back even further. Maybe I have another one. We talked about homologous chromosomes here. So when they replicate with DNA replication, we end up with 2. And we inherit one from mom, one from dad. So when we look at these sister chromatids, you're going to get, like, let's say the red represents the pair of chromosomes from mom, and A is the chromosomes from dad. So we're going to get one of these as a result of meiosis, and they come together at fertilization, and then you have 2 copies of those alleles. So every cell has 2 copies of an allele, and those will separate during meiosis into sperm and eggs, and each getting 1 allele. All right, so let's go back to our slide. So homozygous, they're the same. That can be recessive or dominant. Heterozygous means they are different. So here's homologous chromosomes that we would inherit. Let's say this one is from mom, this one is from dad. Our cells have 1 of each, so we have 2 alleles in all of our body cells. But then when we undergo meiosis, we get just one of those separating into sperm and egg, because homologous chromosomes separate during meiosis. So genotype, remember, is looking at the allele combination. So we can't see someone's genotype by looking at them. For example, they may express the dominant trait, but we don't know if they're heterozygous or homozygous. The phenotype, we can give a little information because it's the physical appearance. So if I see a tall plant, I know that that plant has a big T allele. But again, I don't know if it's homozygous dominant or heterozygous. I'd have to do some test crosses to figure out what that genotype of that tall plant would be. So phenotype starts with PH, think physical appearance. And genotype is genetics, what alleles are present that I can't necessarily see by looking at that individual. Okay, so a dihybrid cross, like I said, uses two different traits in a plant. And it gets a little more complicated then because you're looking at more traits and more allele possibilities or in the gametes. So when we talk about another law that he came up with, he has the law of independent assortment. And what that means is during meiosis, those chromosomes can line up in any independent random way along the middle of the cell when they're separating in the next phase, which is anaphase, and those different gametes can get a different combination of alleles. So again, we can look at the F1 and F2 generation when we do a dihybrid cross. So if we look at this example, we're looking at two traits now. We're looking at tall, and we're also looking at color of the pod. So the color of the pod here is green, that's dominant. So big G is the color of this pod, and little g is yellow. That's the color of the recessive form of this trait, which is yellow. So that's little g. So we have a short plant. Remember, little t means short. So we have a short plant with yellow pods crossed by a tall plant with green pods. Now we know that the alleles are going to separate during meiosis, and we're only going to get one allele per sperm or egg. So all the sperm or egg in this case are going to inherit the T and the G, big T, big G, and all the sperm and egg from this plant are going to contain little t, little g. So when they come together in the first generation, the F1 generation, we have homozygous dominant parent crossed with homozygous recessive parent. We're always going to get heterozygous offspring. So the F generation is always going to have one of each of these parents' alleles, and they're all going to be heterozygous, and they're all going to look like the dominant parent, right? Because they're heterozygous, they contain the dominant allele, they're all going to be heterozygous. But the F2 generation gets a little tricky now because when these alleles separate during meiosis, if we look at the different combinations, one of the sperm or egg could get the big T and the big G, another one could get the big T and the little g, another one could get little t big G, and another sperm or egg could get little t little g. So we have to put all the sperm and egg possibilities along the side here. So this represents the egg combinations. This would be the sperm combinations, which would be the same because the parents are the same. They're all heterozygous for tall and green. But then if I combine those different alleles, I'm going to get quite a variety of different plants. And that's what we see here. We're going to see 9, when I look physically at the plants and I look at the phenotypic ratio, I'm going to see 9 tall green plants, green pod, 3 tall with yellow pods, 3 short with green pods, and only 1 with a short plant with yellow pods. So the phenotypic ratio in a dihybrid cross is going to be 9 to 3 to 3 to 1. And notice that the number of squares we get is 16 because we've got 4 different egg possibilities, we've got 4 sperm possibilities, and you multiply the probability of each together, we're going to get 16 different possible offspring combinations. So if I go back to this and I look at the phenotypic ratio of a dihybrid cross of, in this case we're looking at this, and again, F1 offspring, we're looking at tall green heterozygous parents. We're going to get a 9 to 3 to 3 to 1. So we're going to have, looking at combinations again, we're going to have 9 tall green. We're going to have 3 tall yellow. We're going to have 3 short green and 1 short yellow. And it's again the phenotypic ratio of a dihybrid cross. So again, independent assortment just means that when these chromosomes replicate and come together, they're going to line up on that metaphase plate in a different combination. It's independent assortment, and the segregation just means those alleles separate during meiosis to get all these different cells with different genetic combinations. And that's the role of meiosis, if you remember that from our last unit, is to get reproductive variation. We don't want all of our organisms to be the same because, again, we're going to learn when we study ecology that natural selection favors certain traits depending on what the environmental conditions are. So by having this genetic variability, we have different offspring with different traits and different abilities to survive and use different habitats and all those kinds of factors we're going to talk about. But anyway, so these are all the different egg and sperm combinations that we can see from one cell. And again, this is meiosis 1, we get these 2 cells, and after meiosis 2 we get these 4 cells, again with half the number of chromosomes, but quite a bit of different variability in those alleles that they inherit, not necessarily inherit, but that they form as a result of meiosis. So lots of different combinations, and it demonstrates the law of independent assortment, again, which is how they line up on the metaphase plate, and then segregation is how those alleles separate into the different 4 cells that result from meiosis. So the Punnett Square we talked about is where we take the sperm genotypes, we line them up on one side of the square, and we take the egg genotypes and line them up on the other side of the square, and then we just bring them together. So we just did that here in this. This is a Punnett Square using a dihybrid cross. Here's the sperm or the egg possibilities along the side. Here's the sperm possibilities along the side, and then we just crossed. We crossed this egg with this sperm to get this individual, this egg with this sperm to get this individual, and so on and so on. So we can calculate probabilities. So if I know that the chance of inheriting an E is 1/2 and the chance of inheriting a little e is 1/2, I can just multiply these probabilities to figure out the chances of getting both. So it's the chance of getting big E, little e is 1/2 times 1/2, which is 1/4. So you multiply the top numbers, which is 1, 1 times 1 is 1, and the bottom numbers, 2 times 2 is 4. So let's say I had a different number. Let's say I had, in this case we're looking at attached and unattached earlobes. But let's say I had a probability of event A is 1/4, and event B is 1/, let's say, 6. Then the probability of A and B occurring together, I cannot type for the life of me, is 1/4 times 1/6, which equals 1/24. So just multiply the numbers together, and that's what we can do when we predict the inheriting of offspring. Again, but this is probability, this is not reality, so we have to keep that in perspective. Just because we say an event is going to happen doesn't mean it's going to happen, right? For example, we have the probability of getting a genetic disease, and they might say it's 1 in 100. Well, some people have a child with a genetic disease in their very first child, the very first birth. For that child, instead of it being 1 in 100, it was 1 in 1. It was 100%. So we have to really keep things in perspective and understand when we're talking about families and children, we don't have hundreds and hundreds of kids. So the inheriting of characteristics doesn't necessarily follow probability. It gives us an idea of our chances, but it's not always 100% accurate. So we will pick up in the next video talking more about the Punnett Square.